Okay, so we just went over the 5 R's of cellular division, right? Now we're going to go over binary fission, and there are 3 steps, simple steps, to binary fission. Replicate, segregate, cytokinesis. See, I almost spelled cytokinesis wrong. Yeah, that happened. Alright, so let's start. Binary fission. So, who exactly does binary fission? Let's say prokaryotes with 1 chromosome and circular DNA. That's it. Like E. coli. Ta-da! Alright! First step, replicate. So, replication starts at, let me explain this here. So here we've got a cell. This is the cell membrane around this front here. And then we've got this squiggly mass in the middle. This has several names. It is the nucleoid region. It is not a nucleus. There is no nucleus in prokaryotic cells, okay? So it is the nucleoid region. It is a chromosome. It's an unraveled chromosome. Or it's DNA. They're all the same thing in this case, okay? So we're going to call it a chromosome for right now, okay? So this is our chromosome, unraveled chromosome, and our replication starts at this pink dot. Now what this pink dot is called is the, not the dry, because my O is a little flat on one side, it's the ORI, O-R-I, ORI. And that stands for the origin of replication, okay? That is where replication originates. Ta-da! So smart. All right. So what happens then is there is a complex of proteins that sit on top of this sequence. When a signal is received by the cell or within the cell saying, hey, you need to replicate, this protein complex comes over and just clamps on to the ORI site, and that's where replication starts. Okay? Thanks to these proteins. It doesn't just magically start replicating. There has to be a complex of proteins sitting on top of this ORI site for replication to even start. Okay? So, replication will move, because it is a circle, it will move in a circle from the origin of replication all the way back to the beginning, presumably the beginning, but it will terminate near the beginning at the termination site. Alright? And that's abbreviated as TER, the TER site. So there's the ORI and the TER. Right? So, as we're replicating along here, and going to reach our termination site, the cell elongates to accommodate for the growing amount of shit inside of this cell. By shit, I mean genetic material. Alright? So here you can see our ORI, and it's about to break off the cell here. It's a very bad drawing, I apologize. Alright? The replication will complete at the termination of replication site, and there we have our parent DNA and our daughter DNA. Okay? So this is the replicated shit. Okay, we're on the second step. So that was kind of like 2 steps, but I just wanted to show you like how they looked from before replication really started to when it was finished replicating, okay? Segregate. Now, I don't know what kind of specifics you need to get in here, but because they are genetic copies of each other, they both have origins of replication. Both the parent and the daughter DNA have origins of replication. And those are denoted by the pink spots there. Now, you see these like little green lines? These little green lines denote these fibrils, or these like fibers that are actually attached to the protein complex that is on the ORI. Alright? And that fiber is pulling each specific ORI, the daughter and the parent, away from the center pole like this. Does that make sense? They're doing like this. I'm not making it straight here. Alright. This process, I think this is really important to know. The process of this fiber coming over and attaching to the ori and pulling it away from the center uses ATP. There is energy used in the segregation process of binary fission. I don't know if you need to know if there's ATP used anywhere else. All I know is that your book says ATP is used here. Okay? These fibers, this I also don't know if you need to know this, but the fibers have the same structure or similar structure to actin and have a very similar function to tubulin, which is the base unit of microtubules. I don't know if you need to know that, but it says it in your book, so that's what you need to know, I guess. All right, segregate. ATP, fibers pulling the ori, ATP, 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 okay? All right, and the final step, cytokinesis. Now, as you can see, we have our parent cell and our daughter cell, and because they are exact replicas of each other, they are genetic clones. Clones. Okay? Cytokinesis is essentially the pinching off of the plasma membrane, and this pinching off is actually caused by the same fibers that were pulling, the same kind of fibers that were pulling the ori in the last step, segregation. And it's kind of like what your book says is the drawstring on shorts, where it just keeps pulling it and pulling it and pulling it until finally it pinches off, and that's cytokinesis. I don't know why, but my memory trick for cytokinesis was if you said it kind of Asian, it sounded like something you'd yell when you're karate chopping something, like cytokinesis! And that's pretty much my racist way of remembering what cytokinesis was. So, yes, that's cytokinesis. That's pretty much binary fission, though. It's the prokaryotic way of dividing using very simple